nature's life cycles, days and seasons, weather and storms. People's cells are also propelled by nature's cycles. Our genes, DNA, are copied to RNA, which in turn is converted to proteins, the building blocks of life, before again being broken down and reused. Jørn Kjems, the recipient of this year's Novo Nordisk Prize, has often deviated from the natural research cycle and has regularly sought out new avenues. Earlier in his career, he became fascinated about a remarkable deviation in the life cycle of cells. More than 90% of the RNA that cells produce is never converted to protein. Instead, it is a key to how our body regulates itself.
So if you look at the DNA in a worm and a human, we actually have approximately the same size genome. We also have approximately the same number of genes. So really what makes the difference between a worm and a human is that the RNA is used in a much more complex way.
Well, I think it's very impressive to think about the scientific journey Jørn Kjeldsen and colleagues have traveled. They have discovered so many unforeseen ways in which RNA regulates the phenotype beyond encoding proteins. And these discoveries are now potentially leading to new ways to diagnose and to treat human diseases.
As a child, Jørn Kjems wanted to take everything apart and then reassemble the bits.
I would always draw things so you could look through them. For instance, a car, I would draw how the wheels were able to drive the car, and then draw the engine and how the petrol was running into the engine and how it was ignited.
Nature has many more structures than cars. By understanding these structures, Jørn Kjems hoped to build new things that could potentially help people someday.
So my research went into some of the more fundamental molecules. At that time, RNA was really kind of getting more into fashion. Earlier it was mostly DNA, but in the 80s people realized that the RNA molecule was actually extremely interesting, and from then on it just took off really until today. It's been exploding with science in RNA.
Previously, RNA was viewed as simply a copy of DNA that is decoded and converted into protein.
At that time, it was realized that the way the proteins were made in the cell was actually based on RNA machinery. So the big complex, as we call the ribosome in cells, are actually composed of mostly RNA.
But while he was investigating the RNAs of ribosomes, something else grabbed his attention.
We started to study RNA splicing in archaebacteria when we suddenly discovered that these RNAs actually had long pieces of RNA that was taken out by scissors and thrown away. And we realized that the RNA actually had a very interesting shape. It was circular rather than linear that most RNA is.
The results landed on the pages of Nature, one of the world's most prestigious journals. Jørn Kjems' RNA research career had been kick-started. For his post-doctoral training, Kiems went first to Harvard and then to MIT, where he worked with Philip Sharp, who later received a Nobel Prize for his discovery of RNA splicing.
It was a very fascinating time, maybe the best time of my science career to work in his lab. He was a fantastic mentor, and I learned a lot. And it was also an extreme example to see how RNA could be used in very, very complex ways.
In Philip Sharp's lab, Jørn Kjems started working on a hot topic of the 1980s and 90s, HIV.
These viruses have a relatively small RNA, but it's actually cut in pieces and put together in so many different ways. So you can express more than 60 different proteins from one single genome. Studying that was like Apollo project, sending people to the moon.
The complicated mechanism makes it difficult for human cells to find out what the virus is doing. And when they do, it is too late. The virus has spread around the body. Soon after, a completely new phenomenon appeared: RNA interference. Tiny fragments of RNA that regulate other RNA.
These RNAs are very short and tiny and has been overlooked for for decades for the same reason. But now suddenly we realize that these small RNAs had an extremely important role. Many cancers and metabolic diseases are really caused by these microRNAs that are deregulated. Now you suddenly had a handle on how you could attack the disease.
His laboratory has also described novel methods for delivering RNA therapeutics, which is seen as the new killer drug in its capacity to regulate malignant genes in a highly controlled fashion.
Since then, Jørn Kjems' laboratory has followed two tracks: basic studies of RNA and application-oriented research on drug delivery. The big breakthrough came rapidly and unexpectedly when postdoc Ebeslot Andersen had a crazy idea. He wanted to make a box for the RNA medicine using DNA origami.
I told him that I was reluctant to think that this will work, but fortunately he was very persistent and he bought all these components that he needed for it. And the nice thing is then you just mix it together and it actually happens by itself. So you don't have to do anything, just looking at it. And it can also do billions of these at the same time in parallel.
The DNA origami from Aarhus became world famous in the article published in Nature.
You can compare it to if you took a car and you dismantled it completely into pieces and you then took all the bits and parts and put them into a big bag, shaked it, and then out a completely assembled car comes out.
Jørn Kjems described having a major discovery on his hands as like being hit by lightning. In 2011, it happened again.
One of my postdocs at that time wanted to look into how RNA was regulated in eukaryotic cells, and he had a really interesting observation with some sequences that didn't match, and therefore he actually could only get it to match if we kind of put them together in circles, and that was kind of a déjà vu for me.
The circular RNA molecules from his own PhD days had suddenly reappeared, but in a completely different setting.
The circles seems to work, at least many of them, to be a sponge. And sponge you can call them because they kind of soak up molecules and so they become not available for other processes. So for instance, microRNAs can be attached to these circular RNAs and thereby be pulled out of the system.
This is a completely new way of regulating microRNA and thus our health, and proved to be a very common phenomenon. Over time, Jørn Kjems may have taken many detours, but now the circle is complete.
I moved into different directions, but I think that's my nature, that I like to explore different directions, and then usually you will find a sweet spot, and then you will move in that direction, and then it's okay that other things are dying out. And that's a bit like life, nature. It's like evolution, some things will sustain and other things will disappear.
Jørn Kjems has focused his research in several highly original fields, including understanding how non-coding RNA contributes to cell maintenance and disease development, with a primary aim of defining new targets for disease intervention. This is truly remarkable and outstanding.
Jørn Kjems views receiving the prize as both a very surprising and long overdue recognition of interdisciplinary research.
I was quite surprised that I got the Novo Nordisk Prize, because I thought it usually would be given to people being very dedicated, working on a very specific topic where they become the best in the world. All the work I've been doing has really been dependent on a lot of people, so it has been a lot of collaborations, and somehow I feel this prize is actually given to a whole big community of collaborative partners.